In this session, we will learn how to view a three-dimensional protein structure using the Protean 3D application provided by DNASTAR. Prerequisites for this session are that you have the Protean 3D module of DNASTAR installed. We have received it through their educational license. It's also important that you identify the protein database or PDB file that you want to use. Protean offers the ability to either work by a direct interface to PDB or to open a downloaded file. It's also important to identify the amino acid sequence region containing the disease-associated variant that you've utilized in other exercises. So for example, in the protein that we have utilized throughout these exercises, CFTR, we've learned that the position at 508 is an important site associated with the cystic fibrosis disease. We can also see that adjacent to 508 is the KENII amino acid sequence pattern. Knowing this will be helpful as we navigate the structure as displayed in the various three-dimensional visualization applications. So let's launch Protean 3D, and we'll use the direct interface option. So for our protein, it's 2PZG is the PDB file. So we'll pick that, and then Protean will open the file, and it displays the structure in 3 dimensions, as you see here, and you can rotate it, you can zoom in. One thing we notice is that there's 2 copies of the protein chain. That can get a little bit complicated, so let's hide one of those. So you go over to the molecule option, and you can, in our case, let's hide everything associated with the B chain. So we'll hide both the protein sequence as well as the ATP molecule, some glycerol molecules, and some magnesium atoms. So that definitely cleans up our view. And then we can go back to the protein and take a closer look. So let's zoom in, and then Protean 3D has a very nice feature where you can see the protein sequence below, and above the protein sequence are annotations showing the secondary structure. So helices are identified with the green coloration that you see there, and then the portions associated with beta sheets have the yellow arrows. So I selected position 508, and you can see that it is right at the beginning or end of a helix region. Now let's do a few other things within this. So for example, we can select the region around it and then change the visualization to spheres to get a feel for the surface view of the particular region associated. Protean 3D doesn't offer a great ability to convert the whole molecule into a different view at the same time. So that is one limitation of this. Okay, let's do a few other things. So for example, let's say that we're interested in learning more about the whether or not the site 508 is close to or far from the nucleotide binding site, which you can see there. So by observing this, we gather that it is actually fairly removed from the site where the ATP molecule is bound. But we can learn about other parts of the protein that are close to that simply by clicking on those regions, and the application displays the numbered positions of anything that we click on. Another nice feature is you can select entire secondary structure features, and the program will highlight those features in the 3-dimensional visualization above. So you can see as we click each of these, the visualization changes to highlight the portions associated with those secondary structure regions. In this module, we've learned how to utilize Protean 3D to visualize protein structures. We have seen a variety of features and capabilities that this application provides, as well as identified a few potential limitations.